Welcome in another video lecture of microscopy. So today we are going to discuss about phase contrast microscopy. So before going to discuss about this, I want to recapitulate the entire thing that we have discussed earlier. So phase contrast microscopy will going to solve the problem that we have faced during the dark field microscopy. So it all starts during the lecture number 2, where we have discussed about Brightfield microscopy. In Brightfield microscopy, we have faced a problem that we cannot visualize any kind of living cell with the help of Brightfield microscopy, because the reason is the cells or any kind of biological specimens are semi-transparent in nature, and as a result, when we are illuminating them with the help of a wide range of light source, the cells are illuminated with the background, and it is approximately at similar instant. Okay, so as a result, we cannot distinguish between which is my cell and which is my background. Under such circumstances, we are using chemical stains to color them, so that we can create a contrast between the cells and the background. Okay, but as it is a chemical color, it causes the destruction of the cell, and as a result, those cells becomes dyes, right? And under such circumstances, we are not visualizing Brightfield microscopy. We are not visualizing cells with the help of Brightfield microscopy. So now scientists starts to think about, so okay, we cannot visualize cells with the help of Brightfield microscopy when they are live. That means we have to think about something that can help us to observe them live. So under such circumstances, they have developed dark field microscopy. So what is the principle of dark field microscopy? So dark field microscopy is very similar. Here we are just installing another important instrument in the total instrument of bright field microscopy. That is, we are installing a annular stop beneath the condenser. So what is the utility of that annular stop? So this annular stop is a opaque disc which has a thin transparent layer. This thin transparent layer only allow a hollow cone of light to pass through the specimen. Okay, and the rest of the light will be retarded by the darker part of the annular stop. Okay, and as a result, the amount of light that we are using in case of bright field light is significant. Bright field microscopy is significantly lowered in dark field microscopy. Okay, and as a result, this low illumination help us to at least visualize the live cells. Okay, but unfortunately, dark field microscopy also faces some problem. So what kind of problem? At such amount of low illumination of light, or low intensity light, this become very difficult to distinguish between which is my cell and which is the impurities, the artifacts. Okay, that means in dark field microscopy, it become very difficult to distinguish which is your cell and which is the artifacts that is due to some error during the specimen preparation. Okay, so under such circumstances, FRITZ ZERNIKE developed a new type of microscopy, that is phase contrast microscopy. This phase contrast microscopy will going to solve the two problems that we faces in case of dark field microscopy. One is obviously artifacts, another one is, as we are using a low intense light, this low intense light causes a relatively, it not give you a too much illuminated image. Okay, so illumination, we are not getting illumination while we are visualizing light samples. So the FRITZ ZERNIKE's phase contrast microscopy, because of which he was awarded with Nobel Prize in 1953, will going to provide us a way to distinguish between the cells and artifacts very carefully. And not only that, it also provide us the opportunity to visualize live cell in a very bright way. That means it also provide the brightness, as well as it also provide the differences in the between the artifacts and the cell. Okay, now so let's discuss about on the principle on which the phase contrast microscope works. So the first principle is a very common optical phenomenon that we are going to discuss. So let's go to a diagram that will help us to understand about that optical phenomenon. So the phase contrast microscopy technique is based on the fact that our light have a wave nature. Okay, I hope you have learnt about the wave nature and particle nature of any electromagnetic radiation, and light is nothing but a electromagnetic radiation, right? So it should have its wave nature. Okay, so and another important thing, the whenever we are there are two waves from two different light source, they may be in phase or out of phase. Okay, so what is the meaning of those in phase and out of phase? We have to first learn. Okay, one thing I want to clear you, if two light source, if two wave of two different light source are in phase, then they will together generate a more illuminated light. Okay, that means the intensity of both the light will merge together. Okay, result in the reinforcement or relative brightness of the light. Okay, so when it will happen, let's see. Suppose this is the two wave of two different light source. Okay, so when, and now I want to clear you another important thing, that these two peak of wavelength are so peak of waves. Suppose let's consider this is a wave. So wave, remember, consists of one wave is consist of one crest or peak and one trough. So crest meaning those peaks are called crest. Okay, I have written here, and these bottom parts are called trough. Okay, so the distance between two crest or the distance between two trough is called the lambda, that is our wavelength. So now let's consider this is two wave from two different light source. When the two crest or peak of two waves coming from two different light source will coincides, they will facilitate the reinforcement or the relative brightness by merging together. Okay, so this kind of interference is known as constructive interference, which is the in phase. That means, let again one more time. So this is the two wave from two different light source. Okay, so when the two peak or valleys, they are also known as valleys, coincides together, they together will interact and form a merged light. This merged light has a relatively higher brightness. Okay, so this kind of interference are known as constructive interference, because they are constructing a relative brightness. Okay, and this kind of situation are called as in phase. Now another important thing is similar situation, two wave from two different light source. So if the crest or peak coincides with the trough, they will merge together to form or to destroy the intensity generated by them individually. Okay, as you can see, it is a relatively lower intensity, right, and they will causes relative darkness. Okay, so this kind of interference, as they are destroying their intensity, so this kind of interference is known as destructive interference, and it will called as out of phase. Okay, now let's go back to the PDF. So that is what I have written here, that if the wave peak of light rays from one source coincides with the wave peak of light rays from another source, the rays interact to produce a reinforcement, that is the relative brightness, constructive interference. Okay, however, wave peak from one light source can also interact with the trough of another light source. Under such circumstance, the wave interact to produce the interference which is known as which will facilitate the relative darkness, called destructive interference. Okay, so we will going to use this destructive interference to create a contrast that will help us to brightly watch live cells. Let's see how. So remember one thing very carefully, a phase contrast microscope will going to use the differences in refractive index and cell density of a specimen, okay, to convert those differences into easily detectable variation in intensity. Okay, so here I want to clear another important thing, that in the previous video lecture that was about dark field microscopy, we have faced another important problem, that is dark field microscopy provide us the visualization of a live cell. Okay, there is no doubt about this, but the dark field microscopy will only provide us the outer structure of the cell. We cannot visualize what is inside very clearly with the help of dark field microscopy. So let me introduce that phase contrast microscopy will also going to solve this problem. Let's see how. So here, so if I exclude this instrument, then you can resemble that the entire instrument is dark field for dark field microscopy, right? That means in phase contrast microscopy we are installing another instrument, that is phase plate. Okay, so mind this very carefully, that Brightfield microscopy consists of a light source, a condenser, a specimen, a objective and a ocular. Okay, the dark field microscopy will contain a objective lens extra. Okay, and when we will talk about a phase contrast microscopy, it will have a face plate extra. Okay, so I hope it is clear to you now. So what is the utility of the face plate? So from the light source, light will be travel. So this transparent, let me zoom this picture. So this transparent part of the annular ring will again allow only the hollow cone of light to pass through the specimen. Okay, now this hollow cone of light will be condensed by the condenser as it is its normal work, and the condenser will help to transfer that light to the specimen. Okay, now let's try to think about this. The refractive index of the vacuum or the air is 1, right? There is no doubt about this. And the pure water, in case of pure water, the refractive index is 1.33. Okay, so now let's try to think that the light rays that are traveling for the specimen will go through a relatively denser medium, because there it have to face cytoplasm, it have to face nucleus, it have to face mitochondria stuffs. Okay, so the relative density of the cytoplasm or the nucleus or the mitochondria will be higher than a vacuum, right? So the relatively denser medium will causes those light to bend, okay, that is those dotted lines, okay, and some of the light will also pass through without any diffraction or without any bending. Those lights are this one, okay, that means they are traveling through a part of the slide, let's say. They are not facing any kind of specimen, they are just passed through the slide, or they are passed through the specimen whose whose refractive index or density is not too much as compared to air, okay, and as a result they are not diffracted or they are not undeviated. So these kinds of light will be called as undeviated or unretracted light, and the light who are bent due to facing the relatively denser medium will be known as retarded light rays, okay. And another important thing I want to mention, that these retarded light rays will be retarded by 1/4 wavelength, okay. So again, so annular diaphragm will transfer the hollow cone of light, okay. This hollow cone of light will be condensed by the condenser and transferred to the specimen. In the specimen, due to the relatively increased density of the various kind of cellular materials and the increase in refractive index causes the light to bend, okay. Not only that, it will also causes the light to be retarded by 1/4 wavelength, okay. Now, so we have two kind of light now. One is the undeviated light and another one is the deviated light, okay. So deviated light is retarded by 1/4 wavelength. Now this deviated light will focus to form the specimen by traveling to the objective, then the ocular lens, and then to our eyes, okay. But the undeviated light, after passing through the objective and becoming magnified, it will hit the face ring of the faceplate, okay. As you can see, they are hitting. So the face ring, which is an optical disc of the faceplate, will be constructed in such a way that they will also provide another 1/4 wavelength advancement, okay. That means what? That means our undeviated light was already 1/4 wavelength ahead, and here by using the face ring we are also providing them a extra 1/4 wavelength lead, and as a result the undeviated light and the deviated light will be in 1/2 wavelength out of phase. Remember it very carefully, that as a result, due to the incorporation of the faceplate, the deviated light and undeviated light will become 1/2 wavelength out of phase. Now what is the meaning of that out of phase? We have discussed that they will going to construct a destructive interference, okay. And now when those two kind of light will meet, they will construct the image, okay. So here the undeviated light will going to form the background and the deviated light will going to form the specimen. So the background here will be brighter and the specimen generated by the deviated light will be darker, okay. Now the question is, how this destructive interference causes this difference in color. that how background become darker and how the sample become brighter. Sorry, I have spelled it opposite, that how the field, that is our background, become darker, and how, sorry, extremely sorry, how the field or how the background become brighter and how the specimen become darker. That is, now we have to understand how diffractive interference, destructive interference helping us to create a darker image but brighter background. So let me discuss. So that is the entire thing, that before facing the specimen, they are all traveling at a same wavelength, okay, but when they faces the specimen, the wave in the middle become retarded by 1/4 wavelength, okay. Now the face plate will provide a extra 1/4 wavelength advancement to the undeviated light, and as a result undeviated light is now leading by 1/2 wavelength, okay. And I have told you that destructive interference or constructive interference occur when either the peak or trough will coincides, right? So as I have told you that in that case destructive interference will occur, that means the undeviated light's peak or undeviated light's trough will interact with deviated light's trough or deviated light's peak, okay. But try to imagine that the undeviated light is already leading by 1/2 wavelength, okay. That means before the occurrence of destructive interference, the undeviated light which will going to generate a bright background will get enough time to create the brighter background, right? Because as they are leading by 1/2 wavelength, that means they have enough time before the destructive interference occur to construct the brighter background. After that the destructive interference will occur, and I have told you that this destructive interference is the cause of darkness, so it will causes the dark specimen appearance, okay. Although it will create a dark specimen, and again the color will be black and white, but it will provide us a far better illumination, and we will get the opportunity to visualize what is inside the cell. That means with the help of phase contrast microscopy you can clearly visualize that yes, it is the inclusion body, yes, it is the nucleus and stuff. Why? Because remember I have told you that when our light will pass through the specimen, depending upon the differences in cell density and the refractive index, the light rays will bend and be retarded by a 1/4 wavelength. Now let's try to think about this, that the density of the nucleus will obviously be higher than the density of the cytoplasm, right? That means the rays that is passing through the nucleus will be retarded more as compared to the ray which passes through the cytoplasm. As a result, after the incidence of phase plate, we will have a picture where the nucleus will be darker in appearance as compared to our cytoplasm, and that is the idea. And that is how, on the basis of the differences in the cell density, we can easily visualize which one is my nucleus, which one is my cytoplasm, which one is my mitochondria, anything, right? That means dark field microscopy will only help us to see, yes, it is the cell and it is live, right? That's it. But phase contrast microscopy helping us to visualize the inside of the cell, that is nucleus or cytoplasm or anything. Okay, so with the help of phase contrast microscopy we can visualize microbial motility or any another bacterial structures, for example endospores, for example inclusion bodies, etc. Okay, so after that we can also incorporate a color filter to improve the image also. So phase contrast microscopy is especially useful for studying microbial motility, determining the shape of the living cell, okay, and detecting bacterial structures such as endospore and inclusions, okay, because they are clearly visible, because they have a significant difference in refractive index with that of the water. Okay, so now we will going to see a specimen under the phase contrast microscope. So okay, so the picture in the left hand side that you will get in dark field microscopy, and the picture in the right hand side is the picture when you will using phase contrast microscopy. So now let's try to think about this, that here the appearance of the cell is not too much significant, right? But here we can easily visualize what is inside it, depending on their differences in the cell density, the difference in the phase helping us to visualize it. Okay, so that is a image for dark field microscopy and that is the image for phase contrast microscopy. And now you can clearly understand that why phase contrast microscopy is far more better for visualizing and for visualizing the inside of a living cell over dark field microscopy. Okay, so that is the end of that lecture. So I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends and subscribe to my channel to get more videos like that.